Welcome back to the deep dive. Today, we're doing something a little bit different. Usually, we take a broad topic and kind of chip away at it until we find the core. But today, today we are wading into some murky waters, literally and figuratively.
We are indeed.
We're talking about the creatures that live inside us.
The parasites.
The parasites. And I know, right off the bat, the immediate reaction for a lot of you listening is usually just a visceral, ew. I mean, I think most of us picture a tapeworm in a jar, or maybe that one time we got food poisoning on vacation.
Right.
But looking at the source material we have today, specifically Chapter 69 of Murray's Medical Microbiology, and then we're supplementing that with Chapter 46 of Jowette's. I was honestly surprised. This doesn't really feel like a biology class. It feels like a crime novel.
Well, it absolutely is a crime novel. You know, in clinical medicine, parasites are often called the great imitators. They are essentially the con artists of the biological world. They don't just show up as one obvious thing. They can look like a tumor, they can look like a stroke, they can look like simple anemia. They are masters of disguise.
That is exactly what drew me in. And that's really why I love how we're structuring this deep dive today. We aren't just going to list bugs in alphabetical order, which, let's be honest, is the fastest way to cure insomnia.
And no one wants that.
Exactly.
Yeah.
So we are following the structure of Murray's Chapter 69. It's a systems-based review. We are going to treat the human body like a map of a city.
Right. We're moving away from the strict biological taxonomy, like arguing about whether something is technically a protozoa or a helminth, and moving toward clinical geography. We call this the real estate concept.
The real estate concept.
Yeah.
I really like that. Break that down for me. Why does the location matter more than the actual species name?
Because parasites are absolutely obsessed with location. If you're a doctor and a patient comes in, you don't start with a list of Latin names in your head.
Oh.
You look at the organ system involved. You ask, who lives in this neighborhood? So if the liver is inflamed, there's a very specific list of suspects known to frequent that area. If the brain is involved, well, it's a totally different list of intruders.
So the diagnosis isn't really about memorizing a textbook. It's about knowing the neighborhood. It's beat cop work.
Precisely. And the second part of that detective work is asking, how did they get in? Was it a mosquito bite? Did you swim in a lake? Did you eat a steak tartare?
Right.
Murray provides the map of the neighborhoods, the clinical syndromes themselves, and Jowett gives us the rap sheets, the specific biological mechanisms and profiles of the culprits.
Okay, well, I want to open these case files. We're going to start with the system that is essentially the highway of the body, the blood and bone marrow. And sitting right at the top of this list, the king of blood parasites, is malaria.
You really cannot talk about parasitic disease without starting with malaria. It is the heavyweight champion. According to the global burden of disease data we see in Jowett's, it's still the #1 killer among parasitic diseases globally. But for a student facing an exam or a clinician trying to save a life, the challenge isn't just knowing it's malaria, it's distinguishing the species.
Because not all malaria is created equal.
Right.
Far from it.
Okay, so before we get into the specific types, I want to talk about the fever. Because in movies, malaria is always shown as this guy sweating through his sheets, shaking uncontrollably. But the text mentions something called the sink. What is that?
This is one of the most diabolical mechanisms in nature. Think about it from the parasite's perspective. You are inside a red blood cell. You are eating the hemoglobin, you're growing, you're dividing.
Sounds cozy.
Right until you run out of room. Eventually, you need to burst out to find new cells to infect. Now, if you burst out alone, just one by one, the body's immune system, the macrophages, the white blood cells will just eat you. You're completely vulnerable out in the open blood.
So there is safety in numbers.
Exactly.
Yeah.
The parasites chemically coordinate their development so that they all rupture their host cells at the exact same time. Billions of them. Wait, how do they tell time? Do they have little microscopic watches or something?
It's chemically regulated, and it's often tied to the host's own circadian rhythms.
That's wild.
It is. But the result is this massive, synchronized explosion of red blood cells. When those cells burst, they release waste products and toxins into the blood all at once.
And the immune system just freaks out.
It panics. It releases a massive wave of cytokines, these chemical alarms. And that cytokine storm is what causes the sudden, violent fever spike.
So the fever isn't the parasite itself, it's the body screaming because millions of cells just exploded at the exact same time.
Correct. And because different species have different life cycle lengths, some take 48 hours to mature, some take 72, the timing of that fever spike actually gives you a massive clinical clue about which killer you're dealing with.
That is fascinating. Okay, let's look at the rap sheet for the big bad. Plasmodium falciparum. Why is this one the real killer?
If you remember one thing about P. falciparum, remember that it is totally indiscriminate. Most malaria parasites are picky eaters. They only like young red blood cells, or they only like old ones.
But falciparum.
Falciparum doesn't care. It invades red blood cells of all ages, stem cells, mature cells, dying cells. It takes them all.
So it's just total war in the bloodstream.
Which means the level of parasites in the blood, the parasitemia, can be incredibly high. We're talking up to 50% of your red blood cells infected.
Half your blood.
Half your blood. But the mechanism that actually kills you, this is where it gets really nasty. Jawitz highlights that falciparum makes the red blood cells sticky.
Sticky. Like glue.
I think Velcro. The infected cells actually develop these little structural protrusions on their surface called knobs, appropriately enough, and these knobs contain proteins that stick to the endothelium, which is the lining of your blood vessels.
Why would the parasite want to do that, though? I thought it wanted to flow through the blood to spread.
It wants to avoid the spleen. The spleen is the body's blood filter. It crushes deformed or infected cells. So by sticking to the walls of the blood vessels, the parasite avoids getting washed into the spleen. It just anchors itself in the microvasculature.
But if they're all sticking to the walls...
They clump. They form log jams. Imagine a 10-car pileup in your capillaries.
Oh, man. And if that pileup happens in the brain...
You get cerebral malaria, seizures, coma, rapid death. If it happens in the kidneys, you get black water fever. That's massive destruction of blood cells, leading to dark, black urine and total renal failure.
Yikes. Okay, so falciparum is the sticky killer that clogs the pipes. Now, if I am a student looking down a microscope or I'm a lab tech, how do I know it's falciparum and not one of the milder ones?
Look for the banana.
The banana.
It's the classic visual anchor for exams. The gametocytes, which is the sexual stage of the parasite, are shaped like a curved banana or a crescent moon. No other malaria species looks like that. And the other clue is the double ring. Because the infection is so chaotic and aggressive, you might actually see multiple little ring-shaped parasites inside a single red blood cell.
Okay, so banana and double ring equals the killer, falciparum. Now let's pivot to the ones you called the sleepers, Plasmodium vivax and Plasmodium ovale. Why sleepers?
These two are fascinating because they have a secret weapon. They can enter the liver cells and just stop. They go totally dormant. They become what we call hypnozoites.
Hypnozoite. That sounds like hypnosis.
It refers to sleep. Imagine you get bit by a mosquito. You get sick. You go to the doctor. You take the standard blood stage malaria drugs, and you feel better. You go home. You think you're completely cured.
Right.
2 years later, you're sitting on your couch in Chicago watching TV, and suddenly you have a raging fever and full-blown malaria symptoms.
The sleeper cell woke up.
Exactly. The hypnozoites reactivate. And this is a crucial exam differentiator and honestly a crucial clinical point. If you diagnose vivax or ovale, you can't just treat the blood infection. You have to treat the liver too.
You have to clear out the safe house.
Precisely. You need a specific drug, usually primakin, to kill those sleeping ghosts in the liver. If you don't, they will come back.
That is wild. And visually, how do I spot a sleeper under the scope?
Schiffner dots.
Schiffner dots.
Imagine looking at a red blood cell, and it looks like someone has sprinkled red pepper inside it. It looks like aggressive stippling. If you see that stippling effect, you are dealing with vivax or ovale.
Okay, we have the killer, which is the banana. We have the sleepers, which are the pepper. Who is the old-timer?
Plasmodium malaria. We call it the old-timer because it has a very slow, lazy life cycle. It spikes a fever every 72 hours. We call that a quartan fever.
So a much longer gap between the sinks?
Right, and it prefers to invade old senescent red blood cells. Visually, it forms this thick band across the cell. It's usually a milder clinical picture, but it can persist in the blood for a very long time.
Got it. Now, before we leave the blood entirely, we have to talk about the doppelganger. The one that looks like malaria but isn't, babesiosis.
Yes. This is crucial for listeners in the United States, specifically up in the Northeast. If a patient comes into an ER in Boston or New York with classic malaria symptoms, fever, sweats, severe anemia, but they haven't traveled anywhere near the tropics.
They've just been to Nantucket.
Right. If they've been walking in tall grass in New England, you have to think babesia, because it's not mosquito-borne.
It's tick-borne, right? Like Lyme disease.
Exactly. It uses the exact same vector, the Ixodes tick, so you can actually get co-infected with Lyme and babesia at the exact same time, which is just a diagnostic nightmare for a physician.
I can imagine.
But here is the visual anchor to distinguish it from malaria on a blood smear.
The Maltese cross.
The Maltese cross. Inside the red blood cell, the Babesia parasites reproduce and they form a tetrad. It's 4 little rings arranged in a perfect cross shape. Malaria never, ever does that.
So ring form in a traveler returning from Africa, probably malaria. Ring form in a hiker from Massachusetts. Check for the Maltese cross. It's Babesia.
Spot on. And one more vital clinical pearl here. The spleen is the body's primary defense against Babesia. If a patient has had their spleen removed, maybe from a car accident years ago, or they have sickle cell and they get Babesia, it can be fulminant and fatal. The physical filter is just gone.
Okay, let's move to the hemoflagellates, the whips in the blood, as Murray calls them. We're leaving the inside of the red blood cells now and looking at parasites that swim freely in the plasma. We're heading to Africa and South America.
Right. These are the trypanosomes. Let's start with African sleeping sickness, Trypanosoma brucei.
Vector.
The tsetse fly. And unlike the Anopheles mosquito, which tries to be stealthy and painless, the tsetse fly is a brute. The bite hurts. It's deeply painful.
And the mechanism here, honestly reading Geographic, this part absolutely blew my mind. It's called antigenic variation. Can we really unpack that? Because it sounds straight out of science fiction.
It really is the ultimate disguise. Let's use a bank robber analogy to build a mental model here. Imagine a bank robber escaping down the highway in a getaway car. The police, which is your immune system, get a description. Suspect is driving a blue sedan.
Okay.
So the immune system builds specific antibodies to attack the blue sedan.
So the immune system catches up, attacks the blue car.
But just as they close in, the parasite literally sheds its surface coat, the variant surface glycoproteins, or VSG, and grows a completely new one. Suddenly, the robber is driving a red convertible.
And the police have to start all over again.
From scratch. The parasite has over 1,000 genes dedicated just to doing this one thing. It's like a closet with 1,000 different disguises. This leads to a classic relapsing fever profile. You get sick, your body fights back, you feel a bit better, and then the parasite changes its coat, and you get sick all over again.
And eventually it crosses the blood-brain barrier.
Which leads to the sleeping sickness. And this isn't just being a little tired. It's a profound neurological collapse. You see a total reversal of sleep-wake cycles, extreme confusion, somnolence, coma, and eventually death.
Is there a physical warning sign, like before the coma hits?
There's a classic sign on the physical exam called Winterbottom sign. It's a very noticeable swelling of the lymph nodes on the back of the neck. Historically, slave traders actually used to check for this bump. If a captive had it, they knew they would die soon, so they were rejected.
Man, that is grim. Okay, let's hop across the ocean to the Americas. Chagas disease, Trypanosoma cruzi.
This is a completely different beast. Same family, totally different MO. The vector here is the reduvid bug, also known as the kissing bug.
And I'm sorry, but I have to stop you here because the transmission mechanism, I read this section 3 times to make sure I wasn't hallucinating.
It is deeply memorable.
Walk us through it. The bug bites you.
The bug bites you, usually on the face, near the eye, or the mouth while you sleep. That's why it's called the kissing bug. It takes a huge blood meal. But here's the catch. The bite itself does not transmit the parasite.
No.
No. The bug has a very strong gastrocolic reflex. When it eats, it has to make room. So while it is biting you and drinking your blood, it defecates on your face.
It poops on your face.
It poops on your face. And the feces contain the actual infective parasite.
That is just horrifying.
It gets worse. The bite area itches intensely. You wake up, you scratch the bite, and by scratching, you actively smear the infected feces right into the bite wound or into the mucous membranes of your eye. You are literally inoculating yourself with bug poop.
I need a minute. Yeah. Okay. So assuming I survive the gross out factor, what happens next?
You might get a massively swollen eye. That's called a Romagne sign. It's a unilateral, painless swelling of the eyelid. That's the acute phase. But the real danger of Chagas is the chronic phase. We're talking 10, maybe 20 years later.
This is where the mega prefix comes in, right?
Yes. The parasite has a predilection for the nerve plexuses in your organs, the electrical wiring that tells your hollow organs how to move and contract. It destroys those nerves.
So the organs just go limp.
They lose their tone entirely. Imagine a balloon that has been blown up and deflated too many times. It gets floppy. The heart gets floppy and dilated. That's a severe cardiomyopathy. The esophagus gets floppy megaesophagus, so you can't swallow food. The colon gets floppy megacolon, so you get incredibly severe constipation.
So if you are looking at an exam question about a patient from South America with right-sided heart failure and trouble swallowing,
think Chagas. Think the kissing bug.
All right, let's move up the body. We are entering the control center, section 2. the central nervous system, or as I like to call them, the intruders.
This is a terrifying neighborhood. The blood-brain barrier is supposed to be an impenetrable fortress. It keeps toxins out, it keeps bacteria out. But these specific parasites have found back doors.
Let's start with the brain eaters. The free-living amoebae, Naegleria fowleri.
This is the stuff of actual nightmares. It lives in warm, freshwater lakes, ponds, hot springs, especially in the summer. You're swimming, maybe you do a cannonball, and water shoots forcefully up your nose.
The nasal highway.
That is exactly what it is. The parasite penetrates the cribriform plate.
Explain the cribriform plate for us, just so we have the anatomy right.
It's this tiny, sieve-like bone right at the very top of your nasal cavity. It's where your olfactory nerves hang down so you can smell things. It's really the only place in the body where your brain is functionally exposed to the outside world, separated only by a thin layer of mucus and porous bone. Naegleria eats through that bone, climbs the olfactory nerve like a ladder, and goes straight into the frontal lobe.
And once it's in,
it feeds. It causes panprimary amoebic meningoencephalitis. It is explosive. A healthy young person goes swimming on Saturday, gets a severe frontal headache on Monday, fever on Tuesday, confusion on Wednesday, and is often dead by Friday.
Is there any treatment for that?
It's incredibly difficult to treat because it moves so fast and mimics bacterial meningitis early on. Mortality is well over 95%.
Contrast that with acanthamoeba.
Acanthamoeba is the slower, sneakier cousin. It's an opportunist. It usually affects people who are immunocompromised, causing a much slower granulomatous encephalitis. But for the general public, the big risk factor isn't swimming in a lake, it's contact lenses.
Homemade saline solutions.
Exactly. People trying to save money by mixing their own saline with tap water or swimming in a pool with their lenses in. The amoeba gets trapped between the lens and the cornea of the eye. It causes acanthamoeba keratitis, a severe, incredibly painful eye infection that, if left unchecked, can eventually spread to the brain.
Now wear your glasses in the pool.
Yeah.
Got it. Now we need to unpack one of the most confusing topics for students. The pork tapeworm, Taenia solium.
This is the exam trap of the century. You absolutely have to understand the difference between getting the worm and getting the cysts.
Okay, let's role play this because I need to get this straight in my head. Scenario A. I go to a backyard BBQ. I eat a pork chop. It's a bit pink in the middle. It has a little larvae in it. What happens to me?
In scenario A, you ingest the cysticerci, the larval cysts hanging out in the muscle of the pork. The larva wakes up in your stomach acid, passes to your gut, attaches to the intestinal wall, and grows into a very long adult tapeworm. You start shedding eggs in your stool.
Right.
It's gross, yes, but clinically it's usually very mild. You might not even know you have it. You are acting as the definitive host. You just have a worm in your gut.
Okay, scenario A equals worm in gut. Now, scenario B, I am a strict vegetarian. I have never eaten pork in my life. But I eat a strawberry that was picked by someone who has that tapeworm, and they didn't wash their hands after using the bathroom. So I eat the egg.
This is the dangerous scenario. You ingest the egg is programmed to hatch in a pig. It thinks you are a pig. It hatches in your stomach, bores straight through your gut wall, and enters your bloodstream looking for muscle to encystin in. But it gets lost in the human anatomy. It goes to your eyes, it goes to your heart, and critically, it goes to your brain.
And that is neurocysticercosis.
Exactly. The larvae form these fluid-filled cysts in the brain tissue. On an MRI or CT scan, it literally looks like Swiss cheese. Over time, these cysts die and calcify. This causes massive localized inflammation and triggers seizures. In fact, it is a leading cause of acquired epilepsy worldwide, especially in Central and South America.
So let me recap that.
Right.
Eat the undercooked meat, you get the intestinal worm. Eat the poop the egg, you get the brain cysts.
Perfect. That distinction is worth so many points on any board exam.
Okay, one more frequent flyer in the brain. Toxoplasmosis. Toxoplasma gondii.
This is the one pregnant women are always warned about, the classic don't change the cat litter rule.
Right. Why the cat, though?
The cat is the definitive host. The parasite can only complete its sexual life cycle inside the gut of a feline. The cat then sheds millions of oocysts in its feces. If a pregnant woman ingests those oocysts, maybe from changing the litter box or even just gardening in soil where a neighborhood cat has been, the parasite enters her blood and can easily cross the placenta.
And the result for the fetus?
It's devastating. We look for the classic triad of congenital toxoplasmosis, that's hydrocephaly, which is fluid accumulation and swelling in the brain, chorioretinitis, which is inflammation and scarring of the retina leading to blindness, and intracerebral calcifications.
But there's another population at huge risk for this, right? It's not just babies.
Right. Patients with AIDS. Here's the really scary thing. About 30% of the world's population actively carries toxoplasma right now. It's sitting in our bodies, dormant in little tissue cysts, completely held in check by our immune system.
So we're just walking around with it.
Exactly. But if your CD4 T cell count drops specifically below 100 in an HIV patient, the immune system takes its eye off the ball.
And the parasite wakes up.
It reactivates. In an AIDS patient, it causes massive destructive brain abscesses. On an MRI with contrast, you see these classic ring-enhancing lesions in the brain parenchyma.
So if I see a clinical vignette of an AIDS patient presenting with new seizures and the imaging shows ring-enhancing lesions,
high suspicion for toxoplasmosis.
Moving on, let's head down to the gut, section 3, the intestinal tract. This is incredibly crowded real estate. We can divide this into the microscopic protozoa and the macroscopic helminths, the worms you can actually see with the naked eye.
Let's start with the protozoa. I like to call this the stool profile game. You distinguish these organisms by the type of diarrhea they cause and the patient's specific exposure history.
Okay, let's play. Profile 1. A hiker. He's been camping in the Rockies, drinking from what looked like a pristine, crystal-clear mountain stream. A few weeks later, he has extreme gas, intense bloating, and diarrhea that smells, well, worse than usual.
That's Giardia, the classic beaver fever.
Why is the smell so specific? And the texture. The text repeatedly mentions greasy stools.
The mechanism is the key here. Giardia is not invasive. It doesn't eat your tissue or cause bleeding. Instead, it acts like a wall. The trophozoite form of the parasite has this ventral sucking disc. Think of it like a little microscopic suction cup. It attaches to the villi of your small intestine and essentially carpets the entire wall.
Like putting a tarp over the inside of the intestine.
Exactly. And because the absorptive wall is covered by millions of parasites, you can't absorb nutrients. Specifically, you can't absorb fat. So the fat just passes right through your digestive tract and ends up in the toilet.
Steatorrhea.
Right. Fatty, floating, incredibly foul-smelling stools. If you see the word floaters on an exam vignette, you immediately think Giardia.
Profile 2. Bloody diarrhea. True dysentery. Maybe a traveler returning from a developing nation. Severe abdominal pain and cramping.
This is Entamoeba histolytica. The name actually tells you exactly what it does. Entamoeba means an amoeba inside the gut. Histo means tissue. Lytica means lysis, or breaking down. It is a literal tissue destroyer.
So unlike Giardia, which just sits there like a tarp, this one actively attacks.
It releases enzymes that actively dissolve the intestinal lining, and it eats red blood cells. In fact, if you look at a stool sample under a microscope and you see an amoeba with ingested red blood cells inside its belly, that is pathognomonic. It is 100% entamoeba.
And what about the ulcer pattern?
It digs deep into the mucosa. It creates these characteristic flask-shaped ulcers. They have a very narrow neck at the surface of the gut, but wide destruction underneath in the submucosa. But the real scary complication is if it eats all the way through the bowel wall and into the bloodstream.
Where does it go then?
The portal vein takes it straight up to the liver. It forms an amoebic liver abscess, and the medical description of the pus inside that abscess is just classic, vivid imagery. Anchovy paste.
Anchovy paste pus. I will literally never eat a Caesar salad the same way again. Thanks for that. Okay, profile 3. Intractable, massive watery diarrhea in an AIDS patient.
Cryptosporidium. In a healthy, immunocompetent person, this is mostly a nuisance. Maybe a week of watery diarrhea from swallowing water at a dirty municipal swimming pool. But in an AIDS patient, it is life-threatening. We're talking liters of fluid loss a day. It's functionally like cholera.
And the diagnostic clue for the lab.
The stain. You have to specifically ask the lab for a modified acid-fast stain. The oocysts will turn bright pink or red against a blue background.
Okay, let's upsize. The helminths, the worms.
The monsters.
Yeah.
Worms usually cause disease simply by their physical presence. They're mechanical obstacles. They block things. They suck things.
Let's start with the most common one in the U.S., kids. Itchy butt at night.
Enterobius vermicularis, the pinworm.
Why at night? Is the worm shy?
It's purely strategic. The female worm lives way down in the cecum, deep in the gut. But at night, while the child sleeps and their core temperature drops a bit and the anal sphincter is totally relaxed, she migrates out of the anus to lay her eggs on the perianal skin folds. And the biological glue she uses to stick the eggs to the skin is intensely irritating and itchy.
So the kid scratches in their sleep.
gets the microscopic eggs under their fingernails, touches their mouth the next morning, or touches a toy, another kid touches it, and the fecal-oral cycle just continues endlessly.
And the diagnosis. This is honestly my favorite low-tech medical test of all time.
The Scotch tape test. You literally just stick a piece of clear cellophane tape to the child's anus first thing in the morning, pull it off, and stick it flat on a microscope slide. You'll see the eggs plain as day. They look like little footballs that have been flattened on one side.
Next up, the giant roundworm, Ascaris.
This is the largest intestinal nematode. Think the size of a large earthworm living in your small intestine.
And this one has a bizarre commute, right?
It really does. You eat the microscopic egg from contaminated soil, and it hatches in your gut. But the larva doesn't just stay there and grow up. It actively penetrates the gut wall, gets into the venous blood, and rides the current up to the lungs.
Wait, it goes to the lungs. I thought this was a gutworm.
It is, but it fundamentally requires oxygen to mature to the next larval stage. So it breaks out of the pulmonary capillaries into the air sacs, the alveoli. You get a cough. You cough it up your windpipe to your throat. You swallow it again, and then it goes back down to the gut to finally become an adult worm.
That is just a massive amount of effort.
It is.
Yeah.
During that lung phase, you can get a transient pneumonitis with very high eosinophils in the blood. That's called Loeffler's syndrome. But the adult worm, the real physical danger is a bolus.
A bolus.
A knot. If a child has a heavy burden, say hundreds of these worms, they can physically tangle together into a tight ball the size of a baseball or a grapefruit. It can completely block the ileum, causing a surgical bowel obstruction. Or a single adult worm can wander up in the biliary tree and block the bile duct or the pancreatic duct.
Just a mechanical nightmare. Okay, let's talk about the vampires. The hookworms. Ancylostoma and Decatur.
These enter the body very differently. You don't eat them, they eat you. Or rather, they actively penetrate your intact skin. This is the exact reason public health officials tell people to wear shoes in rural tropical areas.
Walking barefoot.
The infective faleriform larvae live in the moist, warm soil. They sense the vibration and the warmth of your barefoot. They burrow straight through the skin, causing an intensely itchy rash called ground itch. Then they do the exact same lung migration as Ascaris. Travel to the lungs, get coughed up, get swallowed, and attached to the gut wall. But once they are in the gut, they don't just sit there absorbing nutrients. They have specialized cutting plates or teeth.
Teeth.
Literal microscopic teeth. They bite deep into the intestinal villi and they actively suck your blood. They even produce an anticoagulant peptide to keep the blood flowing freely.
So the primary clinical sign isn't really diarrhea or stomach pain, it's blood loss.
Exactly right. It's profound iron deficiency anemia. If you see a clinical scenario with a child in a rural tropical setting with severe microcytic anemia, extreme pallor, fatigue, pica, where they crave eating dirt, and there's no obvious bleeding source, you absolutely have to check their stool for hookworm eggs. They are being slowly drained from the inside.
Man. Next is Trichuris, the whipworm.
Shaped exactly like an old-fashioned buggy whip. It has a very thin, thread-like front end that it sews into the mucosa and a thick back end that hangs out in the lumen. The unique complication here is rectal prolapse.
Rectal prolapse.
In really heavy infections, the local irritation in the colon is so severe that the child constantly strains to defecate. It's called tenesmus. The straining is so forceful that the rectum actually turns inside out and physically protrudes from the body. It's incredibly frightening for parents to see, obviously.
I bet. And finally, for the gut, the one that completely breaks the rules, strongyloids.
The threadworm.
This is hands down the most dangerous worm for anyone who is immunocompromised. It has a unique biological superpower called autoinfection. Explain that, because usually worms need to leave the body to reproduce and complete their cycle right.
Right. Usually the egg has to leave in the feces, mature in the soil for days or weeks, and then infect the next person. But strongyloids' eggs can hatch into rhabditiform larvae right inside the human intestine. And those larvae can rapidly molt into the infective faleriform stage and immediately repenetrate the gut wall or the perianal skin to reinfect the exact same host.
So you act as your own constant reservoir.
Exactly. The infection can persist for decades without any new external exposure. There were American veterans from World War II who served in the Pacific Theater, and they were found with active strongyloids infections 50 years later. They're mostly asymptomatic that whole time.
So what's the trigger? What's the danger?
The danger is when a doctor gives that patient systemic steroids. Say they get a bad asthma exacerbation or a new autoimmune disease, and you put them on a high dose of prednisone.
Because prednisone heavily suppresses the immune system.
Specifically, it dampens the eosinophil and T-cell response that was quietly keeping that worm population in check for decades. Suddenly, the brakes are off, the worms go absolutely crazy, they reproduce uncontrollably, you get what's called hyperinfection syndrome. Millions of larvae rapidly migrating everywhere, lungs, brain, liver, and as they punch through the bowel wall, they carry enteric gut bacteria on their backs right into the bloodstream, causing massive gram-negative sepsis.
So the takeaway is, before starting high-dose steroids in a patient who has lived in an endemic tropical area,
Check a serology for strongyloids. It is literally a life-saving test to run.
Okay, let's move out of the gut. Section 4, tissue, skin, and lymphatics. Let's talk about the things that block the pipes. Filariasis.
Wuchereria bancrofti and Brugia malae. These are transmitted by mosquitoes. But unlike malaria, where the action is in the blood, the adult worms here don't live in the blood. They specifically seek out and live in the lymphatic vessels, the fluid drainage system of the body.
And if you block the drain,
You get a massive backup, lymphedema, fluid buildup in the extremities. Over years and decades of chronic inflammation, this leads to elephantiasis, which is massive, woody, completely irreversible swelling of the legs or the scrotum. The skin becomes severely thickened and hyperkeratotic, feeling almost like an elephant's hide.
how do you even diagnose it early on?
You look for the microfilaria, the baby worms, swimming in the peripheral blood, but here's the biological trick you have to know. Nocturnal periodicity.
They only come out at night.
The adult worms stay hidden deep in the lymphatics, but they dynamically release the babies into the blood circulation specifically between 10 p.m. and 2 a.m.
Why then?
to perfectly match the feeding habits of the specific mosquito vector. It's evolutionary synchronization at its finest. If you draw the patient's blood at noon, it will be completely clear you won't find them. You have to be a bit of a vampire yourself. You have to admit the patient and draw their blood at midnight to make the diagnosis.
Wow. Next, the creature in the muscle. Trichinella spiralis.
This one comes from eating undercooked pork or wild game. Bear meat is actually a classic high-yield exam source in the U.S.
Bear meat. People eat bear.
Hunters do. They make it into jerky or sausage. If it's not cooked to a high enough temperature, you eat the encysted larva. The larvae hatch in your stomach acid, but they don't stay in the gut. They penetrate the mucosa and migrate systemically to your muscle, specifically highly active striated muscle, your tongue, your diaphragm, your calves.
And they just stay there.
They do something incredible. They chemically hijack the human muscle cell's DNA. They force the muscle cell to stop acting like a muscle and transform into a nurse cell. It becomes a protective, calcified capsule that physically feeds and shelters the worm for years. They build a bunker inside your own flesh.
That is pure sci-fi body horror. What are the clinical symptoms while that's happening?
Severe muscle pain, or myalgia, obviously, as they tear into the tissue, but also a very specific, weird clinical sign. Periorbital edema.
Puffy eyes.
Yes. Intense swelling around the eyes, combined with muscle pain, a slight fever, and very high eosinophils in the blood, all happening a couple of weeks after a wild game feast or eating homemade sausage. That is the classic triad for trichinella.
Finally, for the section, the sandfly diseases. Leishmaniasis.
This is a complex spectrum disease. The presentation totally depends on the species of Leishmania and the immune status of the host, but universally, the parasite lives inside human macrophages.
The very cell supposed to eat and kill it.
Exactly. It neutralizes the macrophage's acid phagolysosom. Under the microscope in a tissue biopsy, you see these tiny little amastigotes packed inside the cells. They look like little dots with a dash right next to them. That's the nucleus and the rod-shaped kinetoplast.
There are three main clinical flavors of this, right?
Right. Flavor 1 is cutaneous leishmaniasis, caused by Leishmania tropica or mexicana. You get a nasty, non-healing ulcer with raised, rolled edges right at the site of the sandfly bite. It's often historically called the Baghdad boil or an oriental sore. It heals eventually after months, leaving a severe depressed scar.
Flavor 2.
Mucocutaneous. Leishmania brasiliensis. This is the terrifying one found mostly in South America. It starts as a skin lesion, but months or years later, the parasites actively migrate to the mucous membranes of the face. The nose, the mouth, the throat.
And it eats the cartilage?
It completely destroys it. It can eat away the entire nasal septum and the palate. The clinical term for it is aspundia. It causes truly horrific, permanent facial disfigurement.
Now the third flavor.
Visceral leishmaniasis, also known as Kala-Azar. This is caused by Leishmania donovani. Here, the parasite doesn't stay in the skin. It attacks the deep reticuloendothelial organs, the spleen, the liver, and the bone marrow.
Resulting in what symptoms?
Massive hepatosplenomegaly. You get a spleen that is so engorged it can fill the entire left side of the belly. And severe pancytopenia, meaning all your blood cell lines crash because the bone marrow is physically packed full of parasites instead of making blood. The skin can turn a dark, ashen gray color, which is why the Hindi name Kala Azar translates to black fever. Without aggressive systemic treatment, visceral leash is almost always fatal.
Wow. Okay, we have covered a huge amount of ground here. We've been to the blood, the brain, the gut, and the skin. Let's hit section 5, summary and patterns. We really need to tie this all together for everyone listening. What are the big underlying why patterns?
I think there are two main conceptual models to take away. First is the host pathogen chess match.
Go on. Let's unpack that.
Every single parasite we discussed today has evolved a highly sophisticated strategy just to survive our immune system. Trypanosoma constantly changes its surface license plates to fool the police. Leishmania actively hides inside the cop car, the macrophage. Trichinella builds a calcified bunker in the muscle tissue. Strongyloids clones itself internally without ever leaving the body. They aren't just passively infecting us, they are actively outsmarting our biology at every step.
That makes a lot of sense. And the second pattern, this is the one for the test takers out there.
The eosinophil flag. This is a massive diagnostic clue. Eosinophils are the specific white blood cells equipped to fight large worms. They dump toxic granules onto them. But students very often fall into the trap of thinking all parasites cause eosinophilia.
And that's a trap.
It's a huge trap. The microscopic protozoa things like malaria, giardia, the amoebae usually do not cause any significant eosinophilia. They elicit a different cellular immune response. Helminths, the worms, absolutely do, but mostly, and this is the key, mostly when they're actively invading tissue.
So ascaris migrating through the lungs.
High eosinophils.
Hookworm larvae burrowing through the skin of your foot.
Very high eosinophils.
But an adult tapeworm just sitting happily in the lumen of the gut.
Usually totally normal eosinophils. Because it's not breaking tissue, the immune system barely sees it. It's the tissue invasion itself that rings the eosinophil alarm bell.
That is a golden rule to remember. Okay, before we get to our final thoughts, we have to test our listeners. We have 8 single best answer recall prompts.
Let's see if you were all listening closely. I will read a brief clinical vignette. You pause, think of the answer, and then we'll reveal the culprit and the mechanism.
Ready? Let's do it.
Question 1. A 4-year-old child presents to the clinic with intense itching around the anus, specifically worse at night. A Scotch tape test reveals microscopic football-shaped eggs flattened on one side. What is the organism?
Take a second.
The answer is Enterobius vermicularis, or the pinworm. The nocturnal itching and the Scotch tape test are the absolute giveaways here. If you missed this one, make sure to review the intestinal nematodes table in Murray.
Question 2. A backpacker returns from a camping trip with foul-smelling, greasy, floating stools. There is no blood present in the stool. What is the organism?
Think about it.
That would be Giardia lamblia. The key words there are greasy and floating, which indicates statorrhea or severe fat malabsorption due to those suction discs coating the gut. Check the intestinal protozoa section in Geowets for more on that.
Question 3. A patient with advanced AIDS presents with severe, intractable, watery diarrhea. Stool staining reveals pink, acid-fast oocysts. What is the pathogen?
Got it.
It's Cryptosporidium. This is the classic cause of severe, life-threatening diarrhea in the immunocompromised, and that modified acid-fast stain is your diagnostic key.
Question 4. A patient from South America presents with severe difficulty swallowing megaesophagus and chronic severe constipation megacolon. He vaguely recalls a painless insect bite on his face many years ago. What is the diagnosis?
Lock it in.
That is Chagas disease, caused by Trypanosoma cruzi. The mega-organs are the hallmark of the chronic phase. And don't forget the incredibly gross poop-in-the-bite wound transmission mechanism.
Question 5. A child living in a rural tropical area presents with extreme fatigue and pallor. Labs show a severe iron deficiency anemia. There is a history of walking barefoot in the soil. No bowel obstruction is noted. What is the worm?
Think about the entry point.
It's hookworm, either ancylostoma or nepater. The barefoot entry leads them to the intestine where they actively suck blood, causing that profound microcytic anemia.
Question 6. A returning traveler develops high fevers that spike exactly every 48 hours. A peripheral blood smear shows enlarged red blood cells with intense stippling Schiffner dots. Which specific species of parasite is it?
Narrow it down.
It's Plasmodium vivax or oval. The 48-hour cycle plus the Schaffner dots firmly points you away from falciparum and towards the sleepers. Remember, they need primaquine for the liver phase.
Question 7. A patient presents to the ER with new onset seizures. A CT scan of the brain shows multiple small calcified cysts. The patient admits to frequently eating pork, but the infection mechanism was likely fecal oral. What is the organism?
Tricky one.
Tamenia solium, causing neurocysticercosis. Remember the rule, seizures and brain cysts come from ingesting the eggs from human feces, not the meat. If you ate the meat, you'd just have a gutworm.
Question 8. Last one. A healthy young man develops a rapid, severe frontal headache, high fever, and coma just 2 days after diving into a warm, freshwater lake. He sadly dies shortly after. What is the likely cause?
Final answer.
Naegleria fowleri, the brain-eating amoeba. The history of forcefully swimming in warm, fresh water is the critical clue for primary amoebic meningoencephalitis.
So how did you do? If you stumbled on any of those, I highly recommend going back to Murray chapter 69 and intentionally mapping those clinical symptoms directly to the anatomical real estate.
So we've covered the map today. We've identified all the major suspects. What's the final takeaway synthesis here?
Honestly, diagnosis in parasitology is pure triangulation. You take the geography, where in the world have you been? You take the exposure, what exactly did you eat, drink, or touch? And you take the specific organ symptom. When you layer those three things together, your list of suspects shrinks dramatically from a whole textbook down to just 1 or 2 bugs.
And do you have a final provocative thought for our listeners to mull over?
I do. It's the concept of the silent burden. We spend so much time talking about the acute killers like malaria or Naegleria, but I want you to think about the massive disablement caused by the chronicity of worms. Hookworm causes chronic anemia. In a developing child, that means their brain is deprived of oxygen. They are too tired to walk to school. They are too tired to learn. Their cognitive and physical development is permanently stunted.
Right.
The global burden of disease data shows us very clearly that these ancient, neglected diseases are a massive anchor on the economic and social potential of entire nations. It's not just an interesting medical puzzle for us in the clinic. It's a profound developmental crisis for humanity.
That really puts it in perspective. It's not just microscopic bugs on a slide. It's people's actual lives and futures. Well, thank you so much for diving in with us today. For everyone listening, wash your hands, cook your pork thoroughly, and please wear shoes.
Very sound advice.
We see you next time on The Deep Dive.